My name is Marc Baets. I'm an MD PhD. I was trained at University of Ghent, and I graduated as a specialist in internal medicine, specialized in autoimmunity and also neuroimmunological diseases. I went to Maastricht to become a full professor there, and I'm still working there as a senior professor. Actually, I first started to become a chemist, and I had an uncle of mine who was a clinical chemist, and he said, you have to become a doctor first, and then you can do chemistry more related to medicine. I was, at that age, I was 17 years old, so I followed his advice and started the training in medicine. And then why did I choose the specialty of, I first started to study the specialty of endocrinology because it's the more chemical part of medicine. You have lots of hormones, so the chemistry really inspired me. And then later on, I came on a paper describing a hypothesis on autoimmunity against membrane receptor. The hypothesis was formulated by Vanda Lennon, who was actually an MD, was trained in Australia, and later on went to the US. She never practiced medicine, but she inspired me by her immunopathological ideas. And she first put forward that antibodies against the membrane protein, the membrane receptor, could actually induce a disease. And from there on, I read another paper on the first assay to measure these antibodies, was published by Bernard Rees-Smith in 1974 in The Lancet. And therefore, I wrote, together with the senior investigator at the University of Maastricht, I wrote a project to develop an animal model for Graves' disease. I'm still doing the same type of research, and I'm still working on anti-receptor autoimmunity in Graves' disease, but also against other membrane receptors, being the acetylcholine receptor at the neuromuscular junction of the muscle, and also autoimmune brain diseases, also induced by antibodies against membrane receptors. And why I like these diseases, because the pathological mechanisms are very well known, and there is a specific therapy. So you can actually cure those people. I've done clinical trials, and still I'm an advisor of 5 different pharmaceutical companies working mainly on finding or developing new drugs to specifically treat these type of autoimmune diseases, and mainly myasthenia gravis, because it's a sort of prototype autoimmune disease. As every pharmaceutical company who developed a new compound to suppress the immune system, they first go to myasthenia and then later on to other autoimmune diseases. When I started in the field, there was only cortisone, prednisone, and azathioprine, 2 very powerful immunosuppressive drugs with lots of side effects. Nowadays, there are many new drugs who actually also in a non-specific manner reduce the autoimmune response against receptors. I will give you an example. In myasthenia, there's now a new drug called efgartigimod, Vyvgart, developed by argenx, who actually when you give it to a patient in a few injections, the antibody response goes down by 80% and without any significant side effects. So that's one of the major breakthroughs at the moment. Also, other biological are being developed for these autoimmune diseases. What we discovered and by chance is that, and that was published in Science in 2007, that antibodies against membrane receptors, if they're from the IgG4 type, can undergo FAP arm exchange. That means they become bispecific. So one arm binds to one antigen and the other to the other antigen. And mechanistically, this was a very important discovery. Also, both therapeutically, also is important to, and we showed that if we could make or transform pathogenic antibodies, which are mainly from IgG4 class, to IgG1 class to IgG4, these antibodies become protective against the disease, at least in experimental animal models. As for all of us, it changed since COVID. Before that, we went to the lab every day. Of course, the younger PhD students, they're still in the lab because they have to do experiments. I'm spending my time in between patients. I'm still practicing as a medical doctor, and as a researcher. And I divided my time in the past for 1 day of clinical work and 4 or 5 days a week in more research. And so I'm still doing that a bit less because I'm a senior professor. I'm supposed to be retired, but I'm still working, and I'm not eager to stop, to change my life. Of course, my patients. I mean, seeing patients with autoimmune diseases, also with very rare autoimmune disease. To give you an example, myasthenia gravis, the incidence is about 1 in 10,000 people. And I'm treating more than 300 patients at the moment, using the old drugs, but also using the newer drugs. And what drives me is also to study immunopathological mechanisms. How do these drugs work? Why in some patients a given drug works and why not? And so we have several contracts with the pharmaceutical industry to find out why patients are resistant or are susceptible to the new biologicals. And I think it's important, also money-wise, because you don't want to give a treatment, expensive treatment to a patient where you can predict that it wouldn't work in that particular patient, but maybe another compound might work. So that's one of the main goals at the moment, why I'm still continuing. So patients, but also more than patients alone, translating clinical findings into laboratory work, and also the importance of experimental animal work. Of course, we really restrict that because of all animal rights problems, which I agree with. You shouldn't do an animal experiment if it's not necessary, but for some cases you have to do it. The problem with personal medicine is it becomes very expensive. If only a given drug works only in a single patient, that's, unaffordable, even in rich countries. No, I think personal medicine, what also an important factor is the genetics. We know, of course, the whole human genome now. That's not enough, because we have to study more regulatory mechanism in this human genome. Also, the influence from external agents, from viruses, from toxic agents from the environment, to see what's the effect on the development of certain disease. I'll give you an example. If you have a gene to have lung cancer and you don't smoke, so the chances of you getting lung cancer are very low. That's of course a very straightforward examples, but I think it's also the case for autoimmune diseases, because if you have a certain genetic makeup, a certain HLA type, you're more prone to develop that autoimmune disease, but not spontaneously. There has to be probably an external factor, being a virus or a toxic agent. So the effect of the environment is actually at the epigenetic site, where you can actually influence the expression of certain genes by environmental factors. What I think is very important for a youngster is to, first of all, get interested in science, like I did, by inspiring papers in certain journals. And more in general journals like The Lancet, or more other journals like Scientific American. Read those journals, and also young trainee, and then get inspired. And once you get inspired, try to follow a certain pathway. Don't switch fields every other a year. I started in 78 with anti-receptor autoimmunity, and I'm still working on that, of course. And because the basic mechanisms are there, but the technology is developing in such a fast speed. If when I started immunology, we were talking not about cytokines, but about clonate, supernate, whereas now we have cloned all these molecules. And so that's, definitely much more interesting now. And if you see how the field moved the last 10 years, and when I compare the last 10 years versus the first 30 years when I started in research, we've developed more in the last 10 years than the 30 years before. Everything, because, the ideas we have are not new. We want to look, how does disease develop? But now the technology is so developed, so advanced that we can do it. I think it's pure technology, both, of course, the theoretical aspects of technology, but also practically. And also the collaboration with pharmaceutical companies, and especially in rare diseases. A lot of people opposed to that because they they think we are employed by the pharmaceutical industry. I'm not employed. I'm a completely independent researcher. I get money for contracts, and these contracts, I wrote them myself with my research group, and we get pharmaceutical industry interested. Otherwise, those ideas would never be developed because the money from the government is not enough to answer all these questions. As you know, I'm an ambassador since 1 year, and the reason why I became an ambassador because I had this good relationship directly with the Karger family, and that wake me up to say, okay. And of course, I have another interest in the Karger publication, the book of the anatomy of Vesalius. I bought it years ago, so that's some time ago when I also got in contact with Karger again. Yes, I've been asked to become an editor of a new immunology journal, so I'll be part of that. And of course, I see my role as an ambassador being more senior, and I don't have to work every day to write grants and things. I have more time to think about that and to think about how scientific publications will evolve. Of course, everything you see is online, but I think a good peer review is important. You have to be careful because sometimes some journals have this so-called in-crowd who review the papers, and so that's not good. But I see my role, as an editor, as a reviewer of immunologic papers. In my free time, I'm a collector of art, so I read a lot of books about arts. Another ambition is wine. I'm also a wine collector, so I read a lot about wines, and I like to read novels. A lot of novels. About podcasts, I'm listening to podcasts mainly from the important newspapers, because they can sometimes in 5 minutes they can inspire you, get you the information you want to. But since if you're my age, I'm more in favor of seeing things on paper than to sit behind the screen all day long. Although my screen time is about 2.5 hours a day, so I still do. But my preference goes to reading to books, something you can hold in your hand.